Welcome back, AP Biology, to our discussion on Chapter 9.1. So let's start this off with a question. How did we grow from tiny little babies into the fully grown or maybe almost fully grown individuals that we all are today? From your baby body, did you just stretch into this tall individual? What about when you get an injury? Maybe some cuts and scrapes that leave part of us behind when we do something stupid and get hurt. How do we repair those injuries? Well, the answer to both these questions is cell division. It's how we grow, heal, replace dead cells. Your cells are dying all the time by the millions, and how you create specialized cells for important tasks. Here you can see the cleavage furrows between what started off as a single cell and is dividing into multiple cells to form an embryo to grow into a living creature. Um, in eukaryotic cells, any cell division that results in two identical daughter cells with a full set of chromosomes or DNA is called mitosis. Now, I'm being very particular there that not all cell division is mitosis. And that's because there's different types of cells in our bodies. There's somatic cells, which is most of the cells in your bodies, your muscle tissue, your skin tissue, your blood cells, your epithelial linings, your nervous tissue. All that is somatic cells. They have a full set of DNA. You also produce, through a different process we'll talk about later called meiosis, gametes or sex cells, so eggs and sperm. And those are going to have half the amount of DNA because they're going to fuse together to form a whole. So today we're just talking about somatic cells or mitosis. Left unchecked, however, mitosis is actually the second leading cause of death in the world. So we need to make sure we have good regulatory mechanisms in place. So let's start to explore how all this works. First, let's talk about our DNA. So the whole purpose of life is to pass on your genes. All of your genes, individual genes for making protein, have been carefully selected over years and years, millennia, to be passed on. And so we have many processes to make sure this happens. So first off, we've talked about DNA, those A's, T's, G's, and C's matching up to make the code which makes proteins. So DNA is your code or your instructions. Now, this is going to get wrapped around different proteins called histone proteins to form little octets of histones called nucleosomes, which are going to get wound up again. And basically, DNA needs to be organized and tightly stored. So when you condense your DNA, the word for that is chromatin. Now, most of the time, DNA exists in this state as chromatin. But inside the nucleus, it's still just a mess. It's a big bowl of noodles that's constantly being read and utilized. So chromatin is our condensed form. But when it's time to divide, I don't know if you've ever shared a bowl of ramen with someone and tried to divide it into a second bowl, but it's a mess. Because it's hard to separate that entanglement of noodles. So to make this process a lot easier and a lot faster, we pack our DNA or our chromatin into little suitcases. So we're going to condense that chromatin even more into chromosomes. These will get replicated or copied into this X bar shape. And so we can pass our entire genome or the complete set of DNA into each of the two daughter cells. So you have 23 pairs of chromosomes, and they're numbered based on size from 1 to 22. And then you have your sex chromosomes at the very end, which are either XY or XX. Now, these are not an X-shaped chromosome. These are just single bars, and there's two of them. Because remember, you got one from mom and one from dad. And each individual pair has the same genes. So this chromosome 1 has the same genes as this chromosome 1. Same information, but it could be slightly different alleles. So like brown hair color versus black hair color. So these chromosomes, when they start off in your body, are going to be a single bar shape. When they're replicated or copied, like you see in this little animation here, they become this X shape. Now, like I showed in the last slide, you have 23 pairs. So those pairs are referred to as homologous chromosomes or homologs. So these are matching chromosomes that have the exact same genes on them. Now, remember that homos means the same, so that's telling us that it's the same genes. When these chromosomes are replicated, they're going to be held together by something called a centromere, which is a region of the chromosome that's going to get pulled during cell division, but also hold these homologs together. Now, these two bars are called chromatids. So even though they're each a full chromosome, when they're attached, each individual bar is called an individual chromatid. And because they're identical, they're going to be called sister chromatids. So we have homolog, now a replicated homologs, and then each individual homolog has sister chromatids. I know that's complex and complicated, but going over it now is going to help us process the later steps. All right, that's it for 9.1. I'll see you at 9.2.